Welcome, everybody, to this great class. We've all been looking forward to learning more about plankton and microplastics. Yep.
Why?
A few housekeeping comments before we get started. Just like in Zoom, this system will not pick up multiple voices at the same time. So if you have a question, speak up loudly, and if you would, repeat the question or the gist of it, so people on Zoom, in case they couldn't hear. Try and avoid side conversations, because that will really drive everything nutty. You'll end up with this on the recording. So for the people on Zoom, it's the same thing that you're always used to. It'd be great if you could show your face, but especially if you want to ask a question. Now, Jamie has said that questions are welcome. He doesn't mind being interrupted, so I guess he knows how to skip ahead in that huge deck of his. But just go ahead, if you want to ask questions, don't bother sending it to chat. Just go ahead, show your face, and put your mic on and ask. So with that, it's all yours.
Great. Thanks, Lynn. Well, thank you everybody for coming. My name is Jamie Pierson. I'm an associate professor at the Horn Point Laboratory down in Cambridge, Maryland. How many of you have been there? Visited? Okay, great.
We're coming.
And you're coming, right? Great. If you haven't, I hope you signed up for the field trip.
Right.
Right. Good. So today we're going to talk about from plankton to plastic, the Chesapeake under a microscope. So the plan is I'm going to introduce myself, how I got here. A lot of people say, so how'd you get involved with studying plankton? So I'll give you a very brief history about why I'm doing this. We're going to meet some plankton and some microplastics. I'm going to talk a little bit about how we get this data, like the kind of what we actually do, what a day in the life of a plankton ecologist or microplankton person might look like. And then get into a little bit about what we do know and what we're trying to find out. I'll talk about a couple of projects that I have going on right now.
So you should be able, the people in Zoom, you should be able to see his PowerPoint right now. So if you aren't, send me a text or a chat and I'll be watching.
Great. And oh, you know what? You gotta click back on the PowerPoint. So scroll, yeah, right, just click on it. Perfect. Okay. Now I can advance. Okay. So who am I? I was born and raised in New Jersey, exited on the Turnpike, for those keeping score. I got a bachelor's at University of New Hampshire. I worked as a technician at the University of Rhode Island for 3 years after college. At the Graduate School of Oceanography, I spent a lot of time going out on boats for 2 weeks at a time in the North Atlantic, collecting samples and then coming back and processing those samples. Moved out to the University of Washington, where I got my master's and my Ph.D., and I came to the Eastern Shore in 2006 as a postdoctoral scholar working with then director Mike Roman, who some of you may know. I brought some money with me, and Mike gave me what he called a hunting license, which allowed me to write proposals. And when you start bringing money in, they let you stay. And so I've kind of manufactured jobs along the way for myself. I was research faculty for a few years, and now I'm tenured faculty at the lab. I do quite a bit of teaching, but also continue my research. So I'm going to just give you a little bit about Horn Point Lab and the University of Maryland Center for Environmental Science, because I know a lot of you folks are signed up for the field trip. So we are a research institution and a graduate university for the University System of Maryland. So we have a president that sits at the same level as the president of Salisbury and College Park and Towson and all of the other state universities. We're accredited to provide graduate degrees, not undergraduate degrees. So all of our teaching is at the graduate level, except in the summer we have some undergraduates. So UMCES is a distributed university. We have campuses throughout the state. We have 4 main campuses. The Appalachian Laboratory out in Frostburg, where folks study terrestrial ecology primarily. The Institute of Marine and Environmental Technology in downtown Baltimore at the Columbus, it used to be the Columbus Center, now it's the Rita Caldwell Center. Right next to the aquarium. We have the Chesapeake Biological Laboratory, the oldest marine biological laboratory in the United States. That's in Solomons, primarily fisheries and chemistry. And then over at Horn Point, we have oceanographers and ecologists primarily. So we have a couple of other smaller groups. We work with Maryland Sea Grant, which is a funding body and a group that works with extension agents that help translate science for industry and for management purposes. And then we also have a science communication group called the Integration and Application Network, which is in Annapolis, based in Annapolis. So that's where we are. Oh, you got to click on my.
How many people work in all of these?
So we have about, at Horn Point, at our campus, we have, in the summer, we have about 140 on campus. When it's not the summer, it's closer to between 80 and 100, depending on how many graduate students we have and visiting scholars and things like that. Throughout the whole center, it's probably about 200, spread throughout the entire state. We have about 60 faculty total, and we usually have about 60 or 70 graduate students. And then the rest are technicians, staff, and either office staff or technical staff, and then other folks who maintenance staff. Horn Point, is that next? Yeah. So at Horn Point, we've got 850 acres right on the Choptank River. It's a former DuPont estate. In the early 1970s, the DuPonts donated it to the city of Cambridge. The city of Cambridge went to the state and said, we got all this land for free and we'd love for somebody to put up a university. And they put up a marine lab here, which has become Horn Point Laboratory. So we have a big oyster facility. This is our fairly new solar farm. We've got a 10-acre solar field that produces about half of our electricity. Most of our electricity supports the work of the hatchery, one of the biggest shellfish hatcheries, the biggest shellfish hatchery on the East Coast, I believe. And that grows oysters for restoration, as well as now they sell some oyster larvae for ecology. I'm not going to talk about oysters. I don't do very much with oysters, but I will try to convince some of our oyster folks here to come and talk to you at another time. As I said, this is an aerial view of our campus. It was a DuPont estate, and we have two ram statues at the gates there, and those are they're based on a prize merino sheep from one of the DuPonts. His name is Don Pedro, and he guards the entrance to the lab. So a lot of what we do is work on water quality. We have folks working on restoring water quality from the streams to the bay. At UMCES, we like to say we work from the oceans to from the mountains to the sea. So we have folks studying everything from the deep sea all the way up to the tops of mountains and everything that lives there in between. We also do a lot of teaching. We like to say we are globally eminent and locally relevant. So the work that we do spans the globe, and we bring those lessons back to the Chesapeake Bay to help foster healthy waterways and healthy watersheds here in Chesapeake Bay. Like I said, we're accredited, so our graduate students, we actually have graduation coming up. It'll be in Frostburg this year. It moves around because we have so many campuses. And we'll be graduating, I think, 10 or 12 students this year with masters and PhDs in marine, environmental, and estuarine science. So that's Horn Point and UMCES. Any questions? Okay, so now I'm going to introduce you to some plankton. Before we do that, I just want to make sure that we all know what plankton are. And I do this with every group I talk to because I think a lot of us take for granted that word plankton, right? What does it mean? It's stuff, right? Stuff in the water. So anybody have a definition for plankton?
Pull out your Google.
I'm going to give you one. But
Single cell.
not necessarily. No, but that's part of it. Yeah.
Waterborne? Where?
Waterborne is good. So the term plankton comes from a Greek word, planktos, which means wanderer or drifter. Okay? So they are in the water. They're waterborne. Some of them are single cell. But what makes a plankton is that it can't swim against the currents in the water. Okay, so it wanders or drifts in the water. Now, it's not to say that it can't swim, but the horizontal currents of the water are generally stronger than the vertical currents. And so the organisms that we know as plankton generally can't swim against the current. Fish can. They can swim upstream, right? We think of anadromous fish like salmon and like striped bass and things like that. A tiny little plankton is not going to be able to swim upstream in a river. So they're wanderers or drifters. So why should we care about organisms this small? Well, they form the base of the food chain. And I'm going to get, actually, these slides are out of, where are we? There we go. Okay, so we divide, I apologize, my slides were slightly out of order. So when we talk about plankton, we divide them up in lots of different ways. We can divide them up by what kind of organism they are. So I'll talk about phytoplankton, and phytoplankton are plants. This means that they have chlorophyll and they're going to use the energy of the sun to grow. Okay? We have zooplankton. These are animals. These are going to eat other things. Some of them are single cell, and they can ingest other single cell organisms like bacteria or other protists and things like that. We have bacterial plankton. This is just kind of a broad group of all of the non-nucleus containing single celled organisms out there. We're not going to worry about them too much. We also do size divisions. We divide plankton up by size, and this is really operational, and it goes back to some work in the 1970s by somebody who had a whole bunch of sieves, and he basically poured samples through those sieves, and whatever landed on those sieves, he called them different sizes. So megaplankton are the biggest, macroplankton are the next. Mesoplankton is what I work on. These are about 0.2 to 2 millimeters, so less than about 0.2 millimeters. That would be less than a 16th of an inch or so, somewhere in there. Microplankton are generally single-celled protists. We're going to talk about them a little bit. When you get down into the nano, pico, and femtoplankton, we're talking about bacteria and viruses, primarily. Okay? Most of my work is here, between about 20 microns, so 0.02 millimeters to about 2 millimeters. Okay, so those are the ones that I'm going to talk about. You can actually see some of them with the naked eye if you have a jar. And I meant to bring a jar of plankton, but we had a parasite come in our cultures, and they're not doing really well. And my graduate student has some experiments to do, so I didn't want to disturb the cultures. Okay, so organisms. So phytoplankton and zooplankton, we have our plants and animals. We have lots of sizes, and then we also have life history divisions. So some things are plankton their whole lives. They're not going to grow up to be anything else. Okay? That's the stuff that I work on. And we call them holoplankton. They're plankton their whole lives. We also have meroplankton, which are part-time plankton. This is a lot of the stuff that you all are probably very familiar with. Crabs, oysters, clams, shrimp, lots of fish. They start off life as plankton, so their larvae are tiny little plankton that drift around in the water, and as they grow up, they grow out of that plankton phase and they get bigger. They either settle on the bottom, or they become big enough to swim against the currents, and we no longer call them plankton. Okay? So, let's go back to this. All right, so why do we care about organisms this small? Well, they form the base of the food chain, really. We've got the phytoplankton that take nutrients out of the water, including carbon dioxide, nitrogen, phosphorus, and lots of micronutrients and things like that. They do the same things that plants do. Okay, they just do it in the water. Those things are eaten by zooplankton, and those zooplankton are then eaten by stuff that we care about. Fish, crabs, clams, stuff like that. So if we don't know what's going on here, we don't know really how to get to here. Okay, so that's why we do what we do. When plankton were first described, going back about 150 years ago in Germany, people started looking under the microscope and they started seeing these animals, and then they started seeing these single-celled plants, these things that were kind of green and brown, figured out that they were growing with photosynthesis. We thought that the food chain in the ocean was pretty much as simple as this. It's gotten a lot more complicated over the last few years, and people have identified things we call now the microbial loop. We identified microzooplankton, and I'm going to make it even more complicated in a few minutes. But suffice it to say, when it comes down to it, Zooplankton eat phytoplankton, and fish eat zooplankton in the end. There's a lot of steps in between, and in fact, some recent work looking at some stable isotopes and looking at specific amino acids has shown us that sometimes there's 6 or 7 steps in the food web between phytoplankton and fish. So there's lots of things eating each other in the plankton, and it's something we still haven't really sorted out. We don't know exactly who eats who all the time in the plankton. Because being plankton is kind of terrifying, right? You're in the ocean, you're drifting around, there's nowhere to hide. It's not like being on the savannah, right, where you can run up a tree and hide from a lion, right? There's nowhere to hide. So the way that you die in the plankton, more often than not, is that you get eaten by somebody else. Okay. Oh, I did want to point this out. So I'm introducing plankton. I'm going to talk about microplastics in a little bit. Our definition for microplastics are all pieces of plastic that are smaller than 5 millimeters. A quarter of an inch, something like that. That's the operational definition for microplastics. Folks are starting now to talk about nanoplastics. These are things that are smaller than 0.1 microns. So these are about the size of a bacteria. Okay? What we're going to talk about are microplastics and macroplastics. Bigger and smaller. I'm not going to talk about the really tiny things. We're going to talk about the really small things and then the bigger things a little bit. Okay? I just wanted you to see where those things fit. They don't line up with the, and the point here is they don't necessarily line up with the same definitions we use for plankton. So microplankton are 0.2 microns and smaller. Microplastics are 5 millimeters. Sorry, 0.2 millimeters and smaller. Microplastics are 5 millimeters and smaller. All right. I told you it was going to get a little more complicated. I just like to tell this story because I find it really fascinating. So for a long time, scientists have identified things as either phytoplankton or zooplankton, plants or animals. All well and good. About 30 years ago, and actually a lot of this work was done by Diane Stecker, who is professor emeritus at Horn Point Lab. People started looking at the single-celled organisms that were in the water, and they found out that a lot of the ones that contain chlorophyll, a lot of the ones that are doing photosynthesis, using the sun and nutrients in the water to grow, can actually also eat things. Okay? So when times are bad, say the middle of the winter, or in the middle of summer when they've exhausted all of the nutrients in the water, they can decide to go ahead and eat other single-celled organisms in the water. So they can do both. They're kind of like Venus flytraps. But the interesting thing is, all the single-celled organisms in the water that we normally find, there's really just one group that is only photosynthetic, that is only phytoplankton. All the other groups can do both things. They can eat and they can use the sun. We call them mixotrophs. That one group that only photosynthesizes are called diatoms. And this is a picture, this is almost entirely all diatoms. That was actually taken during my graduate work from Puget Sound. Are any of you gardeners? Do you use diatomaceous earth?
Yeah.
What is it about what makes diatomaceous earth useful for gardening?
Or nutrients.
Do you know what nutrients?
Calcium.
And
Other minerals, I suppose.
yeah, there's a lot of silica.
Yeah.
Right? Okay, so there's a lot of silica in diatomaceous earth. So diatoms are responsible for, depending on the papers you read and how you do the calculations, about 20 to 30% of the photosynthesis on Earth. Okay, so they produce a tremendous amount of oxygen. And what makes them really unique is they can't swim at all, and they have this shell around them that's made out of silica. It's made out of opal, basically. So all these individual dots on here, they're connected. They're individual cells, but they grow in chains, and they're covered in silica. So they have these silica cells. So they're pretty heavy. They produce lots of lipids and things like that, lots of fats to keep them buoyant so they don't all just sink out. different kinds of diet. Anywhere you find water that's exposed to sunlight, you're going to find diatoms pretty much. You're going to find some of them. There's I don't know how many species there are, but there's tons and tons of species.
You said they produce oxygen also,
Yes, through photosynthesis. Yep.
and then migrates up into the environment.
Yeah, it diffuses out. Yep.
Yeah.
Yeah. So they're responsible. Estimates range from 20 to 30% of the oxygen that's produced by photosynthesis on Earth. Those are estimates. We haven't been able to interview all the diatoms on Earth, but those are estimates. Okay. Talked a little bit about sizes. I just want to give you an idea because there's a tremendous variability, and I can put that table up with lots of different numbers on it, but there's a huge variability in sizes in the organisms that we consider plankton. And so I scaled all of these little lines and brought some pictures in from things that we find in the Chesapeake to just give you an idea of the variety of sizes in plankton, okay? And this is hard to do. I don't know if you've ever, I think about, when I think about this, I think about trying to make a scale model of the solar system. And, if the Earth is the size of a golf ball, Jupiter would be, in like Kansas, and it would be the size of a basketball. The size ranges are, I don't know if that's exactly true. But the point is that the scale is really, really big. So these little dots would represent the smallest, this little dot here would represent the smallest of the flagellates, the smallest of the single-celled organisms. The bigger single-celled organisms would be a line about that big. You can see these two dots next to each other. Microplastics are about this size. So this would represent about 5. If this represents about 5 millimeters, these are the dominant animals in the water. So these are multicellular organisms. They're about 1 millimeter long. This line represents that size. And then 1 centimeter would be about our biggest things that we might consider plankton. These are, for example, in the Chesapeake, these would be mysid shrimp. Now, most of the animals I'm going to talk about today are going to be crustaceans, and I'm going to talk a lot about copepods because that's what I study, that's what I know. They are probably the most numerous animal on Earth. And I'm going to talk about why we think that and why that's important. Also, I talked about marroplankton a little bit. These are the things that are, all the other things on here are plankton their whole lives. They don't grow up to be anything else. The marroplankton are things like polychaete worms that live in the sediment. This is a crab. This is a picture of a crab larvae here. So things that are going to grow up to be something else can be, as plankton, they can be lots and lots of different sizes. Okay. Any questions about that stuff? Yeah.
Planktons cannibalize?
Can they what?
Do they cannibalize?
They can, yes. In fact, the copepod that I work on, most of when we do reproduction experiments to see how productive they are. As soon as the female produces eggs, you have to take her out of the jar and put her in a clean jar. If she's not well fed, she'll eat her own eggs. So it's tough being plankton. You got to do what you can to survive.
Hey, your mom.
Okay. I put this in, and I will be perfectly honest because I made this graph and I really liked it. But it was for a textbook, and it's to describe the different patterns of behavior that larval plankton use to keep themselves in an estuary. Okay? And I'm not going to go through all of the details necessarily, but I mentioned that that plankton can't swim against the horizontal currents, but they'll go up and down, right? So they'll go up and down. And in an estuary in particular, like the Chesapeake Bay, we usually have fresh water at the surface flowing towards the ocean, and we have salt water from the ocean flowing in at the bottom. And so plankton can actually use those those currents flowing in different directions to maintain their position in the estuary, just by going up and down and kind of riding a conveyor belt around. And so crabs do this. They actually, the females release their eggs kind of out near the shelf. The larvae actually leave the bay, and then later on, as they get bigger, they ride the currents back in before they settle. You have shrimp in the upper estuary that do kind of the opposite. Well, sorry, they do the same pattern, but they do it in the upper estuary. They release their eggs, and then they go up into, they release their eggs, and these are a little bit backwards because the surface layer should be flowing out of the estuary. But regardless, they use this up into, don't worry about the details, but they use the different flowing currents to keep their position in the estuary or to find places where they want to be for the next phase of their life. So there's really complicated behaviors that all of these little tiny little organisms do. And it's part of what got me interested in this work. When you start looking under the microscope, start seeing all these things alive and realize that there's this whole world that you can't really see going on under the microscope in the with the tiniest of animals out there. It gets kind of fascinating. There's a lot of questions left to be answered. Yes?
How long do these creatures you study live?
Really good question. So the holoplankton, like I study, generally speaking, it depends on the temperature and the size. The larger ones live a little bit longer than the smaller ones. And the warmer it is, the faster they go from egg to adult. Okay, so their generation time goes a lot. So in the summer, the ones that I live, it takes them about a week to 10 days to go from when eggs are produced until they are an adult that can produce new eggs. In the wintertime, it's about 30 days. And they live here in the Chesapeake the whole time. So
How long?
well, I mean, they live all year. If they don't get eaten, they can live for months and months and months.
Oh, okay.
So we kept them, when we keep them happy in the lab, I can keep a female copepod. I think she's, no, okay. Coming up on copepods in a minute. But I can keep a female copepod alive for 3 or 4 months, and every single night she'll produce 20 or 30 new eggs. So in reality, in the Chesapeake Bay, she's probably going to be eaten within a couple of days. So. All right. Okay, so I just want to talk to you. I mentioned mixotrophy. I'm going to just introduce you to some of my favorites before we get into my all-time favorite plankton. Okay. Dinoflagellates are a really diverse group. These are both single-celled organisms. So these are protists. These two groups, dinoflagellates and ciliates, a lot of them have chloroplasts. Almost all of them, if they have chloroplasts, can also eat. So they're almost all mixotrophs. But they're really, really fascinating groups. Dinoflagellates are named because they have 2 flagella. You know what flagella are? Little tails, right? Okay, so they use one for propulsion and they use one for steering. So they can actually direct themselves where they want to go. Lots of dinoflagellates are implicated in red tides or brown tides, toxic or harmful algal blooms and things like that. Lots of them are also bioluminescent. I do a bit of work in Puerto Rico in the bioluminescent bays, and what makes them bioluminescent is a particular species of dinoflagellate. They're really, really fascinating. They're pretty slow growing, and one of the reasons for that is that their genomes are about 10 times the size of ours. So in a single dinoflagellate cell, they have about 10 times as much DNA as we have in one of our cells. They have a huge number of genes, and it gives them a really flexible lifestyle. You can imagine if you're a mixotroph, you would kind of need a lot of machinery because you need to photosynthesize, but you also need to be able to ingest and eat all kinds of things. So that's part of the reason their genomes are so big. But they can also use lots of different, they can metabolize lots of different things, and they can adapt, they're pretty adaptable to lots of different conditions. And that's what gives them these huge genomes. Yes?
Is Pfiesteria plankton?
Yes, Pfiesteria is a dinoflagellate. So Pfiesteria is a harmful algal bloom. It was implicated in the 1980s, in particular, in North Carolina, in a lot of fish kills that were largely attributed to the blooms of Pfiesteria were largely attributed to runoff from a lot of large pig farms and things like that in the Neuse River and in the New River and stuff like that. So it's a very complicated organism. It has lots of different life.
Also here in Maryland.
And yes, it was here in Maryland. Yeah, we called it the hysteria. There were a few years where there was a ton of work being done on it. But yes, I don't have a picture of it, but it's in this same group of organisms here. This one is, that's Alexandrium, or it's either Alexandrium or Pyridinium. They're closely related, but they produce diuretic, they produce saxitoxin, which is diuretic shellfish poisoning. So these things can produce toxins things that are toxic to humans. They can also produce things that are not toxic to humans, but are toxic to other organisms out there. So they use that probably partly as a defense mechanism to keep things from eating them and allow them to bloom. But they're also really fascinating shapes, and they're really, really pretty under the microscope. Fun to look at. So ciliates, so these guys have two flagella. Ciliates have tiny little hairs, they're cilia. They're another really, really diverse group. Some of them have hard shells, like these tintinids here, and some of them don't. They're things like, if you remember from your biology courses, paramecium, right? That kind of crawls around the surfaces, that's a ciliate, okay? What's really neat about ciliate, what I find really neat from a biology point of view with ciliates is they have multiple nuclei in each individual cell. So they have one kind of master nuclei, they have like one big brain, and then they've got all these little brains that do very specific tasks. So they also have very big genomes, not as big as the dinoflagellates, but they've got these other extra little nuclei that can do specific tasks that allow them to be really adapted to their environment. Both of these also have a lot of parasitic forms. So the organisms that I work on, copepods, we find parasitic ciliates on them a lot. You find these they're attached to the outside, they're kind of like stalks. And when you see, that's what happened this week. In fact, why I don't have live copepods in the jar to pass around right now, because we had a whole bunch of ciliates show up in our samples. And we have to do some work to try to keep them healthy for our experiment. All right, so those are all single celled. I'm going to just talk about a couple more zooplankton before I get to my favorite. So these are things that are found in the Chesapeake Bay. Ketogonats, we don't talk about very much. So this is a really weird word, ketogonat, but it means spiny teeth. And the common name for them is arrowworms. They're only about half a centimeter to a centimeter and a half long in the Chesapeake Bay. So let's say no more than half to 3/4 of an inch long, and they're pretty much transparent. They're ambush predators, they're voracious predators. A lot of times when we catch them, they're mostly transparent. You can see other plankton kind of lined up in their guts. But they're found around the world. They're a really diverse group. They're a phylum unto themselves. So they're distantly related to lots of other things, but they're not part of the annelid group or anything else. They're pretty separate from everything. You find them in the deep sea, you find them in the surface ocean, you find them everywhere. And like I said, they're voracious ambush predators. They just kind of hang, they're see-through, they're transparent, they hang in the water, and they wait for something to swim by, and then they lash out and grab it and digest it. And then sea nettles and ctenophores. They get a bad rap, but they're really fascinating. So ctenophores are comb jellies. So these guys don't sting. Okay, and we have a couple species in the Chesapeake Bay. In the wintertime, we have kind of big orange ones called burrowii, which are, they're actually predatory on the other species that we have here, which is Nemiopsis leadii. But what makes ctenophores really cool is they've got these little hairs along the sides of their bodies, and that's what they use to both swim and capture prey. So they move those little hairs, and they actually, if they encounter a prey item like a copepod, these are the copepods, they don't, instead of having stinging cells, they have sticky cells. And so the prey item will stick to the cell, and they'll use those little hairs to kind of pass it down to the mouth and then ingest it in the mouth. The really kind of terrifying thing about ctenophores when we think about it is that they don't seem to ever be satiated or full. So most animals, if you feed them, they get to a point where they're like, you know what, I'm full, I can't eat anymore. We haven't found that point for tinophores. The more you feed them, the more they eat. They're just absolutely voracious predators. Now, they don't have a lot of sensing organisms, so they have to rely on kind of bumping into things. But really the higher the concentration of food they're provided, the more they will ingest.
And what size are they?
So they can be a couple of inches long, but they're not really strong swimmers, and that's why we include them as the plankton. Yeah.
Question.
Yes.
Have you run those through next-gen sequencing to figure out what genes control that kind of behavior?
There are folks, so I don't know the answer to that directly. There's a lot of work on the phylogeny of tinophores, so like how closely different groups are related, but we haven't, I don't know very much about the metabolism work, but that would be something interesting to look at, yeah. Yeah, I don't exactly know how it works, but I just know that all the feeding graphs I've seen just are a straight line. You know, food on the x-axis, ingestion on the y-axis, the more food, it doesn't ever level out.
I can see a practical application where they can start, you check those genes into NFL prospects.
That's right, that's right. Careful what you wish for, though. And then the sea nettles. So these are true jellyfish. That's kind of what we think of them as. They're cnidarians. They're related to sea anemones. They're related to corals and all things like that. They have stinging cells. Recently, a couple of years ago, the one from the Chesapeake Bay was actually, they did a lot of genetics work on that one and identified that it's different than the one on the coast. So the one in the Chesapeake has been renamed. And now I'm blanking on its species name is, I'm blanking on the genus name, but its species name is Chesapeake now. So Chrysaora. So it's Chrysaora chesapeakei. So the one in the Chesapeake has been identified as a separate population, genetically distinct from the ones that are on the coast. These guys are also a little bit opposite. The tinophores seem to be a little bit more tolerant of lower temperatures and lower salinity. The sea nettles have a more complicated life history pattern. So when they release their gametes, their sperm and eggs in the water, they fuse. They need a place to attach to, and they love oyster shells. So there's a connection between sea nettles and oyster shells, and there's also a connection between salinity and sea nettles.
You said sperm and the egg.
Yes.
From the same animal?
No, they're not from different animals.
No, there's male and female.
Yes.
Okay, all right.
Yep. Yeah. And then they attach, they grow, and then they actually can reproduce asexually in the phase that's attached to oyster shells. So they can reproduce asexually, emitting these things called a phyrae, which are really, really cute little baby jellyfish. And they release them into the water, they grow up into males and females, and then there's a sexual phase that then attaches to hard substrate again. It's really complicated. But they require hard substrate. And that's, I think, a really interesting thing. There's some folks have suggested that for a number of years in the Chesapeake, sea nettle numbers were kind of lower than they had been. They've been up the last couple of years. I don't know if any of you have been out in the water, but oh my goodness, you could walk across Chesapeake last year on sea nettles. But it may be related to the amount of oyster shell that's available out there in the water. I'm not saying it is. This is a hypothesis that some folks have proposed. Okay, fine. There we go. Okay, I'm going to talk to you a little bit about copepods. This is my favorite copepod. This is Acartia tonsa. This is from the Chesapeake Bay. Copepods are probably the most numerous animal on Earth. Okay? When I say animal, I'm talking about multicellular animal, not single cell. So these are multicellular animals. The reason we say that is because the Earth is covered by about 70% water, right? 71% water. Anybody know the average depth of the ocean? Or want to hazard a guess? Average depth of the ocean.
feet.
Yeah, I know, it's not something you think about. I'm not expecting anybody to know that.
1,200 feet? Average, I mean.
It's deeper than that.
20,000 leagues.
It's not that deep. Yeah. So, and being, I'm not going to be able to do this easily in my head, but to convert it to imperial units, but it's about 3.7 kilometers, 3,700 meters deep, average depth of the ocean. Okay? So 70% of the Earth, 3.7 kilometers. What's that? It's got to be over two, it's like 2.5 miles, something like that.
13,000 feet.
13,000 feet. I knew you'd help me with that one. So about 13,000 feet. That's a lot of water, right? That's a lot of water, okay? And these animals are found in this group of animals, not this particular species. There's about 16,000 species or so. But they're found anywhere we have water. They're found in high mountain lakes. They're found in streams. They're found in the deepest parts of the ocean. They're found all over the ocean. Yes?
Have they been found in ice?
Yes. And we find their eggs in sediments and things like that. In fact, there's some folks who have been able to take sediments and they bring dormant eggs from copepods that are about 100 years old, and they can wake them up and they can keep going. They've been well preserved. So lots and lots of different forms. Generally speaking, most of them go through about 12 stages in their life. These are all scaled to the exact same size. I had a postdoc named David Elliott who did this work. He was raising them and took pictures of each of the individual stages. So the first 6 stages are called naupliar stages. Those are larvae. There's an egg here. This is about the size of the eggs. This is a female. This is a male. These naupliar stages are kind of like little eggs with legs. They don't swim very well. In fact, they don't start feeding until the 3rd stage. So these 2 stages are just relying on yolk from the mother, from the egg. Then they start feeding, and that's when they start growing. They're crustaceans, and so they molt. Okay? So every time they molt, especially at the young stages, they sometimes they add a leg, sometimes they add a segment here and there. And then after the 6th naupliar stage, they undergo a metamorphosis, and they start to look more like the adults. We call these the copepodid stages. And they start getting big really fast when they get to that stage. So some of the, yes?
What happens to their shells after they molt?
They just sink out. So they're made of chitin. So they sink to the bottom, and there's lots of bacteria. Oh, this gives me good question. So there's some bacteria that can actually metabolize chitin, that can eat it. One of those bacteria is Vibrio cholerae, which is the cholera, the bacteria the pathogen that causes cholera. And a really neat, fun story. I will try not to tell too many stories about plankton. But a neat, fun story is they renamed the Columbus Center, what used to be the Columbus Center, for Rita Caldwell, who was the first female director of NSF, I think, and she was also faculty at Maryland. She's from Maryland, right? Okay, so one of the things that she did was she was a microbiologist. She studied cholera. She realized that copepods, which are much bigger than bacteria, carry that Vibrio cholera on them. So the carapace, the shells of these copepods, are covered in lots of little bacteria. Some of them are the Vibrio cholera, and so she worked with groups, I think especially in India, women who were collecting water in the river in
Ganges.
the Ganges. Thank you. Worked with groups and filtering that water through saris actually reduced the incidence of cholera drastically. And it was getting rid of the cholera, but the reason it was getting rid of the cholera, because the cholera was hanging out on the copepods. So it's a copepod story, even though we think it's a cholera story.
So what is chitin? I've never heard that word.
Okay, I'm going to get in trouble because I don't do biochemistry or chemistry. But I think it's related to things, the kind of stuff like our fingernails and stuff like that. Okay? I don't know specifically. Yes?
Is that related to the Vibrio, the
very little fleshy.
The Vibrio fulnificus or Vibrio parahemolyticus flesh eater, it is related, yes. And they are also, my understanding is they are also chitin, they also feed on chitin. And so similar kind of thing, if you filter the water, you get rid of a lot of that stuff. Yep. All right, a couple of copepod fun facts. There's currently about 14,300 accepted species of copepods. I said 16,000 because there's a lot of genetics work coming out that's showing that there's much more diversity than we really knew. There's lots of forms, parasitic forms, benthic forms, and pelagic or planktonic forms that live free living in the water. They were originally described by Aristotle in one of his papers on natural history. And that's because of the parasitic forms. So if any of you are fishermen or a fisher folk, and you catch a fish that has what we call lice attached to its gills or its eye or something like that, most of those are copepods. So they're big copepods. As in their early stages, in the non-parasitic stages, they're really small. Then they attach to a fish, and they get really big sometimes where we can see them. And Aristotle was the first one we know of who actually described those. I mentioned this, that they're probably the most abundant animal on Earth. And if you want to do the math, we can go through it later, but I generally don't do math in public. Okay, so just switching over to microplastics. Any other, well, any questions about copepods or plankton in general? For now? Yeah.
I keep thinking about how they might fit into the food chain with Homo sapiens. You know, is there some thought about ultimately farming these critters?
Yes, you can actually buy a jar on Amazon of copepods, dried copepod. And the reason is because there's some, I didn't even talk, I mean, I'm trying to temper myself because I could be here for hours. But, so there's a whole group of copepods that actually have an overwintering phase. Okay, so they live multi-year life, they have a multiple year life cycle, and they get to a point in late summer where they realize winter is coming and they pack on lots of lipids. And it can be about 80% of their body weight can be fats and lipids. And then they swim down to the deep water and they go into kind of a hibernation state. And you can see with, for a lot of the copepods that live in the plankton, they have really big, long antennae. And they're not completely asleep, but they go down deep and they just sort of hang in the water like this. And they use all those sensing organisms to make sure if somebody comes by and tries to eat them, they can swim away. So all those little hairs are different sensory organs that allow them to sense chemicals, basically smell, or sense pressure, like a wave of something bigger swimming by. And so there are some there's a couple of researchers in Norway where they have lots of this. Their Calanus is the genus name. There are lots of Calanus in fjords and stuff like that. And in the wintertime, they go and they scoop them up because they're very high. They're long chain fatty acids, and they're these lipids that are really high in omega-3s and things like that. It takes a lot of copepods to make dinner.
I was just gonna say, it looks like the biggest one is like 1.5 millimeters.
Yeah, I mean, the world record is about 16 millimeters, actually, these big bathypelagic ones. But the ones that the dominant copepods, the most abundant ones that are big like this are maybe 5 millimeters, okay?
Oh, okay.
Yeah, and they're, yes, in the winter they have lots and lots of lipids on them. But unintended consequences, whales, so when they go down deep into these layers, they form pretty dense layers. They all kind of aggregate at one layer in the water. And the Navy has known about it because there's a really strong acoustic signal for these things also. Whales can use it, right? So the whales go down and they can come into these fjords and they can just kind of cruise along at a specific depth and eat all of them. So, and to get them to produce those lipids requires a tremendous amount of algae, so I don't know that, I think a lot of people have thought about this and a lot of people have tried. I don't think that there's a market for human consumption necessarily. Yeah.
Shots.
Go butt shots. Yeah. I like that. Yeah, that would work. I won't say I've done that at 3 o'clock in the morning.
Out loud.
But I won't say I didn't either. So. Yeah, you do strange things on research cruises at 3 o'clock in the morning. Right. But they are important, so going back to, so they are important in the human food chain because they're actually really great food for fish. And so they're a little bit harder, they are harder to grow than a lot of the other things like rotifers and like brine shrimp, sea monkeys, right? Sea monkeys are dead easy to grow. The copepods are actually a much better, higher quality food, and so there's a lot of aquaculture groups that are working on growing copepods for aquaculture at the early stages of a lot of those organisms. Okay, I promised I would talk a little bit about plastics, and I'm going to talk a little bit about plastics. So, all right, I mentioned this before. Why are we thinking about microplastics? I know a lot of you have heard about microplastics. You've probably heard about the Great Pacific Garbage Patch, right? It's been in the news a lot. This was from a coordinated beach cleanup across the United States of the top 10 beach cleanup items that were collected. I think this was in 2019. 9 of the 10 were plastic items. Okay, 9 of the 10 garbage items collected on beaches for beach cleanups are plastic. The thing that's not, generally speaking, is usually cigarette butts. So other than cigarette butts, almost everything we pick up off of the beach is, if we're cleaning up beaches, picking up garbage, almost everything is plastic. Microplastics are everything that's smaller than 5 millimeters or about 3/16 of an inch. You've probably seen this little recycling symbol, right? So not all plastic is the same, right? And I found that this is actually a really long infographic that I really, really like. And it comes from, actually it was a small business that put this together, but it talks about how much, all the different types of plastic here. We have PET, high density polyethylene, PVC, low density polyethylene, polypropylene, polystyrene. I'm not a materials chemist. I mean, I can't write these, the formulas out for these things. Just know that they have really different life cycles, right? Some of them can decompose in 5 to 10 years under perfect conditions, under absolutely perfect conditions. PVC pretty much never breaks down, which is why it's really useful for pipes in our home, because you really don't want your pipes to corrode, right, or degrade. But it's really bad when it gets in the environment unchecked and we can't get it out, right? Low density polyethylene, 500 to 1,000 years. That might as well be forever as far as we're concerned, right? So the only, really the PET or PETE is the only one that has like a 5 or 10 year lifespan. The rest of these are 20 to 30, 50 years, things like that. Varying toxicity levels depending on the other types of chemicals that are used with these. And again, I'm not a chemist. I'm going to say that over and over again. So I can't answer questions about the chemistry here. But know that these things are, not all plastic is the same. They vary tremendously in their impact and in their lifespan in particular. And that's why we're talking about them now, because they're persisting in the environment, right? Oh, I'm talking too much. Okay, so just a couple of slides about, so I've shown you all about the plankton. I want to just show you a little bit about how we do the same type of sampling, whether it's plastics or whether it's plankton, okay? We do the same kind of stuff. And I brought an example. In fact, this is the kind of net that we use on small boats here in the Chesapeake. We have, this is and the video that's showing is perspective of sampling. I put a GoPro on one of our nets and sent it down. So this is, if you were hanging onto the net, this is what it would look like as we were deploying the net. It's not important, but I thought it was fun, and I did it, so I brought it.
Want to hand stretching it out?
Sure, thank you. Excellent. So this is the kind of thing we use to collect either plankton or plastics. Now you'll note this is nylon, so it is a plastic. Okay, when we do our plastic work, we have to really think about contamination. We have to really account for it. We do a lot of samples and blanks. So this we can put in the water and we can drag it around. There's usually a bucket on the end. I took it off because it was lighter. You can take the bucket off, pour the sample into a jar and preserve it. Sometimes we bring the sample into the lab and we pick individuals out to do experiments with plankton. The nice thing about working with plastics is that they don't degrade, and so we don't really have to worry too much. Thanks a lot. We don't have to worry too much about how we handle the samples. The plastics are going to be there. The bad thing is they don't degrade. So, we know that the plastics are going to be there. Okay, so we use lots of different things to collect samples. Just wanted to give you an idea. These are all pictures from the Hugh R. Sharp, which is the boat that's out of Delaware. It's a 150, 160 foot research vessel. It's run by the National Science Foundation, but it's based at the University of Delaware. This is an electronically controlled net. The rest that we can actually sample different depths with it, and we can measure temperature and salinity and oxygen and chlorophyll. We can take lots of measurements while we're taking our samples. This is called a CTD rosette. And these bottles are open at both ends, and we can it's again electronically controlled. You can put it in the water, and you can close the bottles at particular depths so that we can sample very specific depth horizons for lots of different things. This is what it looks like when we get a sample. Lots and lots of little tiny things. Fish usually can avoid our nets. As we said, they can swim better than the plankton. The plankton can't really avoid our nets. So we pretty much, once in a while, you'll get a fish that's been stunned or is really unhappy in your nets. But this is mostly what it looks like. Sometimes you get one big jellyfish, and then we take lots of pictures with it. But generally speaking, this is what the samples look like, whether they're plankton samples or whether they're plastic samples. Like I said, we do the same thing. So I got involved in the plastic stuff because I've been doing this for years. I've always ignored the plastics and just tried to look at the stuff that was alive. Now we're trying to ignore the stuff that's alive and look at the plastics so we can figure out what the impact is out there. We get our samples. That's Mike Roman. I don't know if any of you know Mike. He was former director at Horn Point, my mentor. Sometimes we do experiments with our plankton. We call this the plankton wheel. This put bottles on there, it rotates very slowly to keep everything in suspension, kind of mimic being out in the ocean. We use these to measure rates and processes. So we can count all the stuff in the samples, right? We can just count and we get an idea of how much is out there. But if we want to know what they're eating, how much they're reproducing, things like that, we do experiments back in the lab. Or on the ship. These are on the ship. This is down in Chesapeake where we put the bottles back in the water so that so it mimicked natural conditions. This is in the lab at Horn Point. So this is kind of a day in the life. This is a good day in the life. This is when I'm not doing paperwork. But this is a good day in the life of a plankton ecologist. Okay. I'm kind of running out of time, so I'm probably going to have to come back another day. We're going to do choose your own adventure, if that's okay, because I don't want to keep you here too late. We can either talk about the dead zone in the Chesapeake Bay, or we can talk a little bit more about plastics. Anyone have any? The dead zone is low oxygen water. It's more of a plankton story. If you want to hear more about plastics, we can do more about plastics. Okay, want to vote for dead zone? The dead zone? Okay. All right, I apologize I didn't get more to plastics, but they're inert and they're going to be there. Okay, so I'm going to talk a little bit about the dead zone. So this is some work that I've been doing for a while. Has anybody heard of the dead zone? Yeah, in the Chesapeake, we have them. It's also been identified. The two big ones in the United States are really the Chesapeake and the Gulf of Mexico. This is a map from a paper that was out a few years ago. So the dead zone is low oxygen water in the ocean or in coasts or in estuaries. So it's low oxygen water. It happens in the deeper parts of the water. And anybody know why that is? Why we don't have low oxygen up near the surface generally?
Circulation.
Yes, both of those things, circulation. So there's two ways to get oxygen into the water. There's two ways to get oxygen into the water. One is our second best friends, other than the copepods, the phytoplankton, that are producing oxygen through photosynthesis, right? And that requires sunlight. So that has to be near the surface. So photosynthesis produces oxygen. So all the phytoplankton producing oxygen in the surface layer. The other way is through mixing of the wind. So you churn up the water and you can inject bubbles into the water. It injects oxygen into the water.
Won't the environmental oxygen diffuse at some,
Yes, but
If
not
they
not very quickly. Yes.
okay.
Mhm.
So not enough to get a decent, you
Yeah.
need the other events going on.
Right. The only way to get it into the deep water is through mixing it down and through diffusion, which is a very slow process compared to photosynthesis and wind mixing at the surface.
So is there an explanation as to why this is existing in North America and also in Europe? It's
Do you have an explanation as to why it might exist in Europe and North America primarily?
probably something to do with
I don't know if you can read that.
population density.
Right. So the prevalence of these dead zones or hypoxic zones is strongly related, not only, but strongly related to population density. Okay, where there's a lot of humans, we generally get a lot of hypoxia. You'll notice there's a lot of humans in this part of the world and we don't see a lot of that, right? That is probably more related to reporting and to the amount of data that's available, right?
China.
There's other things that affect this. Places like the Chesapeake is a long, narrow channel. Circulation is not as strong as it might be in other places. It's also kind of shallow compared to some other estuaries like Puget Sound and things like that. So the bathymetry, the coastline, the shape, all those things contribute to how much low oxygen we have. But generally speaking, hypoxia is driven by excess production. Increased nutrients into the water causes things to grow, and then they die, and they sink out and they die. And then they...
Agricultural practices as well.
Yes, agricultural practices. So increased nutrients either from urban runoff or agricultural practices and things like that. It's all excess nutrients. You're giving these plants, they're growing really fast to the point that the rest of the plankton can't really keep up, plankton and whatever's in the water can't keep up eating them. And so then they die and they sink out. And then you have the bacteria that come along and decompose them, right? And that decomposition process uses oxygen because it's respiration like we do, right? We use oxygen. I'm using a lot as I yammer on and on here. But it uses oxygen and it doesn't replace it. And so that's what happens with this hypoxia.
And what about the CO2 production?
That's, yes, as we're using oxygen, we're producing CO2 in the water. That's correct. Yep.
Does that affect anything?
Well, it's going to affect the pH, but that's a whole other week-long course. So, yes.
More acidic?
Yes.
Yeah.
Yeah, the increase in CO2 is going to affect it's going to affect the buffering capacity of seawater, and it's going to affect the pH of the water.
Lower
Yes.
pH.
So can't you break up a dead zone by stirring it up?
Sure. If you've got a lot, like think about let's go.
Well, there was a big tip.
This is the dead zone in the Chesapeake Bay. You'd need a lot of swizzle sticks.
A lot of boats going
Right?
racing back and forth.
Well, and they'd have to be deep, right? Because this dead zone, all right, this is going to be hard to see. So this is depth over here. This is what the dead zone looks like if you took a cross section of the Chesapeake. Okay, so this is depth. The surface is the straight line. So you can see that the dead zone starts at about 10 meters, which is about 30 feet. So you'd have to have a minimum 30 or 40 foot long swizzle stick to mix that up. Yeah. So this surface layer, we call this the surface mixed layer, because this is the layer that's mixed up, the surface part of the ocean that's mixed up by the wind, the part that has the connection to the atmosphere. We call it the surface mixed layer in oceanography. And then this would be this is our oxycline or pictocline or thermocline. So cline means gradient. So this is the place where you have a switch, okay, where you have a big gradient between the surface water and the deep water, okay? And I didn't know I was going to do physical oceanography today, but that's okay. I love it. When you have a really strong difference, especially in an estuary, you have really fresh water at the surface and you've got salty water down the bottom. And there's a big difference in density between that surface water, which is why it stays on the surface. That fresh water is lighter in density. It's like oil, and think of it as like oil and water. And those different densities create a really strong layer that's very hard to penetrate. Okay? And so that's why that's why it's hard for oxygen to get down into that deep layer. That's part of the reason.
Too many thoughts.
So is the dead zone only in the place where there's a channel? We went to Poplar Island last week. And most of the Chesapeake, the average depth is only 21 feet.
That's right.
So it's the dead zone that's down where the ship channel is.
Yes, that's
Okay,
exactly right.
That's what I need to
Yep.
grasp.
Yes, that's a really, really good point. Now, it sometimes, depending on wind conditions and things like that, it can spill up onto the shallow water. But you're right, and I don't have a good graph of it. The Chesapeake has these really shallow shoals, and then it has this deep channel in the middle. And that's, I think, largely, and Doug is the geologist, so he's going to correct me when I'm wrong, but the Chesapeake is really the drowned river valley of the Susquehanna River, right? And so it was the river during the Ice Age in particular.
No, right there, that's the thalweg of the Susquehanna. Thalweg is the deepest, fastest flow of the river, and that's the concentration where the anoxic or the hypoxia occurs. It doesn't occur in the shallow parts, which makes the oysters very happy.
Right.
And in the fish, if they get into that, they swim away. So it doesn't affect the fish.
Right. But the oysters can't really get away, so they just can't live down.
And they don't.
And does it go up the rivers too? It looks like it's
The
going up
hypoxic, yes.
Yeah, it follows those deep channels.
So it follows the depth of the water.
Yeah, it's trapped. Yeah, so what happens is you've got this excess, you got a lot of excess production, it sinks down, and then you have decomposition that uses up the oxygen. So you still have excess production over those shallow areas, but they're more in contact with the surface because they're getting mixed up.
So the ships going through aren't enough.
They're not enough. Nope. The Chesapeake, this is really big. I think we forget about the scale until you're out there in your 15-foot Boston whaler and you look up, right? But, this is 300 kilometers. So that's, this is 160 some odd miles, I think, from the dam at the Conowingo Dam down to the mouth. So the scale is really, really big. These comments about mixing it up. So these things are happening. In fact, I have a colleague over at Chesapeake Biological Lab. She's working with a group in Annapolis. There's a small creek, and I can't remember which one it is, but they're putting aerators in that creek because they had a lot of water quality issues, and they're using it at my colleagues, the homeowners pooled their money and decided that they wanted to try this. And my colleagues are actually getting to do the experiment. Like how much do they help? How much do you have to aerate? Yeah, I think it's on the Severn. Yeah. It's Laura Harris is doing it. Jeremy Testa.
It's like the collection ponds. We put bubblers in those to keep the water circulating.
At Easton Club. Yeah, at Easton Club. Right. And there's also for a while you had the floating. There were
Yes, we had the plants,
Right.
I don't know what you call it, and they ate a lot of it,
yes. Mm-hmm.
so.
Right. They're using up. They're going through a lot of those nutrients.
Well,
Are we happy?
We'll stay on for half an hour.
Okay. So how about I do a few more minutes? Well, but keep interrupting. Yeah.
Can I go back to a plastics question?
Sure, can you just talk louder so people can hear you on Zoom?
Sorry.
Maybe come up here.
I'll repeat it.
Okay.
What is the origin and provenance of the smaller plastics in the Chesapeake Bay.
Okay. It's almost like you're a plant, but you're not. Okay, so going to the plastics, let me skip ahead first. Do plankton eat plastic is one of the questions. I'm gonna, yes, plankton do eat plastics, they don't like it. Now I'm gonna go back to your question. They can't live on it, right? They'll eat it, but, all right. So I'll just speak for a few minutes about plastics since we're here. And I'm happy to answer more questions about the dead zone, and I apologize that I ran out of time, but I get excited. So.
You got plenty of time, dude.
Okay. Let's see, here we go. All right, so we're doing a couple of things in the Choptank River. We're doing a couple of things in the Choptank River. It's a new project. This is a map of the Choptank. The red lines are where our stations are. And the question from Doug was, where do the small plastics come from? We're trying to figure that out, and we're doing it in two ways. We're doing it by sampling above the wastewater treatment plant for Talbot County, which comes in just below the Dover Bridge. We're sampling above it and then below it. So the idea being that if there are plastics coming from that wastewater treatment plant, we'll be getting above them, and then we'll also be getting below them. So if the hypothesis is they're coming from there, we should have much lower numbers at our furthest upstream station and higher numbers at our lower station. The other thing we're trying to, yeah?
Is there a lot of tidal, or is it pretty much flowing at that point?
Very good question. We are above, so we figured out approximately what the tidal prism is, so about how far the tide moves, and we're above that.
You're above.
So the mean flow, the average flow of that river is downstream. The tide could push stuff up, but it is eventually going to move down the stream. So we're sampling above the tidal prism that the plastic shouldn't be carried, nothing should be carried as far up as we're sampling. Should. Doesn't mean it's not gonna, but that's the plan. Okay, so there may be stuff coming in from there. We're also doing degradation experiments. So we've got a couple of these, actually, we got permits from MDE to put these out in the system. 6 different types of plastics, our 6 main types of plastics. These are little strips. They're pre-weighed, so you know exactly how much they weigh. And then we put them out subtidal and intertidal. And we want to see how quickly they break down. And because that's the other place they might be coming from. They might be coming from plastics that are trapped in the marshes or in the sediments that are breaking down and releasing those microplastics into the water. Okay? I don't have any data to show you just yet. We started this project last year. We're still working on some of the details of how we're going to do our sample processing. But we're looking into whether most of the plastics in our samples come from the wastewater treatment plant, or if they come from intrinsic sources, plastic bags, water bottles, things like that are trapped in the marsh and that are breaking down over time. So we have 2 of these on the roof of one of our buildings. Those are our controls, how plastics break down just sitting out in the environment. We have a couple of them in the water. I will say that we've done some of this work already in, my colleague William Narden has done some in acetate, and we've done some in the cove that we have at Horn Point Lab. Because one of my concerns is, well, aren't we just putting plastics?
Can we report you?
These are not breaking down very fast. Okay, so it's not like we come back and there's nothing left. Right? We're talking very, very small changes in weight over time with these plastics. But we are learning a lot about how the different types of plastics, we're doing it in we're looking for really small changes. So we're able to see how these different types of plastics are behaving, whether they're completely under the water or whether they're intertidal, so wetted and dried twice a day, right? The other thing we're doing, and I'm not allowed to do this, William doesn't let me touch his drone, but William is flying, and I think I might have a drone picture. No, I don't have a drone picture, but we're flying drones over our study sites, And the drone has a infrared camera on it, and using that infrared camera, you can actually, it's got two cameras, in fact. It's got this crazy fixed wing drone. This isn't like the little ones that you can get at Walmart. This is this big fixed wing drone. It can cover about 200 acres. And it's got two cameras on it, and we can actually differentiate plastic from the images that it takes. And the idea is, at least for some of the big pieces, we can look at those images over time. And it's got centimeter scale. So we're not looking at microplastics, but we're looking at macroplastics and whether or not they're breaking down or disappearing in the marshes over time.
You're also assessing what plastics, how they get stored in the marsh?
Yes, so that is exactly, yes, good question. So the purpose of this project, we're asking three main questions. Is how does the amount of microplastic change with season and location in the river? So I already mentioned we're sampling above and below the wastewater treatment plant. So we're sampling down there. We're sampling in all 4 seasons. We actually went out in February and took some samples this year. We're going to be going out in spring here pretty soon as I can schedule it, as soon as the wind dies down. We're looking at how different types of plastic break down in the river. And then William is also building a model. So, and when I say sampling, let me step back. When we're sampling, we're not just taking water, we're taking water samples, we're taking sediment samples, and we're sampling in the middle of the river as well as on the edges, okay? Because one of the big questions that we have is how, that NOAA, who's funding this work, is really interested in is what is the fate and the transport of plastics through coastal systems? We hear a lot about the Great Pacific Garbage Patch, but there's no plastic plant in the middle of the Pacific. All that plastic had to come from somewhere, right? So it ultimately came from a land-based source. It was produced on land and it had to get out there. And so one of the big questions is, how does all that stuff get out there? And how does plastic move through our coastal systems before it gets to these big garbage patches? And that's what we're trying to address using the Choptank River as an example or as a case study. So the Choptank, this is not a high density region, but it's right in our backyard. And we know the Choptank really well. And we have really good numerical models that can predict flow rates, that we can kind of play games with. We can put different marshes, we can put different seagrass beds in, and we're trying to understand how when you put plastics in that river, how they move through the river under different conditions, different flow conditions, different seasons, and with different levels of marsh activity.
Do fiberglass hulls contribute? You have 500,000 of them. What would you?
They probably do. And one of the things that we're just adding to our project, we have a new microscope that we raised with philanthropic money and from foundation money. In fact, the last 2 pieces supposedly were delivered today, so I'll get them tomorrow to the lab. So this is a shortwave infrared microscope, and when we find particles in our samples, we're going to be able to look at them under this microscope, and using a little bit of math, a little bit of statistics, and looking at the reflectance of each particle at different wavelengths in the shortwave infrared between 900 and 1700 nanometers, you can actually differentiate types of plastic. And so fiberglass is what we can put fiberglass particles under that scope and see how they see where they fall in this spectrum, right? And we should be able to determine that. I don't know how many we're going to find. We may find tons, we may not find a lot. I imagine we might find a lot of fibers. Polyester nylon fibers is a really big thing these days that a lot of the plastics folks are thinking about and talking about. We may find lots of synthetic rubber particles from farm equipment and things like that. We may find lots of plastic bags. We may find lots of, hopefully we'll find less, right? Easton has banned them. I know I've got a whole bunch of reusable bags in my car that I still forget every time I go to tackle. But But they're in my car now.
I think that'll take a while, but and it's only Talbot County.
Yes, that's true. And it probably will take a while,
That's right.
but what's really cool is we were out there last year, we're there this year. Hopefully, if we can keep this going, we may be able to detect a change. And I think what's neat, the original proposal that we put in was just going to count the total number of plastics, but with this new instrument, hopefully we'll be able to pinpoint the type of plastic. And if we can do that, we might be able to say, over time, the amount of plastic bags may have decreased. Like that's a hypothesis that we can now test. We can go out and test if we continue collecting these data.
And then people can all stop complaining.
I don't know that people ever stop complaining.
That's true.
Present company not excluded. I will complain plenty.
Jamie, is there anything else worldwide comparable to the collection in the Pacific of plastic?
Isn't there
Yeah,
smoke
yes.
in the Atlantic?
Okay, I wish I had you know what, I do have a global map, don't I? All right, I didn't know I was going to be teaching global oceanography, but I love it. I'm by training an oceanographer, so oceanographers view the ocean as a system, and I'm a biological oceanographer, so I study plankton, but I have to think of plankton within the whole ocean system, right? So I have to know enough about physics to be dangerous, and enough about chemistry to be dangerous, and all these things. So that's why I get really excited about this. And I don't have all of my arrows and things, but in all of these major open ocean spaces, okay, north or south of the equator, we have a gyre. And this is a whole nother seminar I'd absolutely love to give on global ocean circulation or something to that effect. But it all has to do with wind and the rotation of the earth and Coriolis. There are gyres. There's these big giant eddies that are set up you guys know the word gyre? No. Okay, so a gyre is a big circular ocean circulation pattern. Okay? GYRE. Okay? So and it's set up, the Gulf Stream, right? Have everybody heard of the Gulf Stream? So the Gulf Stream runs north along the east coast of the United States, then makes a right turn. It goes across. It becomes, for a whole variety of reasons I can talk about another time, it becomes a little bit weaker, but then it comes down the coast of Africa and comes back across the Atlantic Ocean to meet up again with the Gulf Stream. So there's this big circular gyre. It flows the opposite way in the south, okay? And that has to do with Coriolis, which is the force that's driven by the rotation of the Earth. Okay, we have a similar pattern in the North Pacific. There's a Kuroshio current that goes up over here, runs across, comes back down along California, Mexico, and then comes back. So there are these big ocean gyres. And in the middle of them, you have what we call a convergence zone, where everything collects. So the Great Pacific Garbage Patch gets all the press, but these gyres occur in all of our oceans where we have a lot of big, above or below the equator, north or south of the equator, where you've got a lot of big open area. And those, in all of our oceans, it collects lots of things. In the North Atlantic, that's called part of that's the Sargasso Sea. You've heard of Sargasso, right? And Sargasso is big news in Florida, and I do a lot of work in Puerto Rico. They really worry about when the Sargasso comes in, but there's lots and lots of Sargasso out here in the middle of the Atlantic, and a lot of it is collected together because of that big circular motion in that. So does that answer your question? Yes.
Just to add, if you're concerned about the collection of plastics on the coastlines, if you've been to Cancun or Oba or places like that, you cannot walk on the beach without stepping on it. That's because what they do is they release their trash just offshore. And so the plastics are coming by wave and coastal circulation.
Right. Growing up in New Jersey, when I was little, there was a lot of trash on the beach. And there were a lot of jokes about it. And there was...
Still are.
Yeah. Yeah. But there's not as much trash. There's still a lot of jokes, but there's not as much trash. There's definitely not. Because they shut down one of the big dumping sites. And it really didn't take that long. It was really within a couple of years that the amount of trash really decreased along the New Jersey beaches. Yeah.
In the Nature magazine article yesterday.
Oh, yes.
They described the Great Pacific Garbage Patch as being 120 square miles. And they also indicated that there are a lot of coastal critters that have found their way to this huge area. And maybe you can help us put that scale into perspective of how much 620 square miles, 620,000 square miles actually is. But so these critters have made an adaptation. What's that ultimately, or what kind of inferences can scientists make about how that's going to impact the ocean food chain?
Okay, can you summarize for the?
Sorry. That's right. Okay, there was a question. There was an article yesterday in Nature, Ecology, and Evolution where they examined the communities, the organismal communities, live plants and animals that have arisen around in particular, in this case, the Great Pacific Garbage Patch. Okay. And one of the things that they did was they looked at all the different organisms that were found on pieces of garbage that were floating around in the Great Pacific Garbage Patch. They identified them, they collected these garbage, they took the bugs off, and they looked at them under a microscope and identified everything, and then they classified them as being oceanic. So meaning they're primarily found offshore, right? They're not something you would find on the beach necessarily. Or whether they're coastal, whether they're things that originated from coastal communities. One of the things they found, the two things that I took from that paper, and I thought I had a slide in here that had data from that paper, but I don't. So there were two big takeaways for me. One was that there were lots and lots of coastal organisms on those pieces of garbage in the middle of the Pacific Ocean. So they don't normally make their living there, okay? But they were making their living there. And the conclusion from that by these authors is that this is a potential way of exchanging organisms across very long distances that wasn't present before we had all of these plastics.
Microplastic boats.
Yes, that's exactly right. And last week I got to go to Guam for a conference and for a meeting with some folks that I do, and it was a conference on island sustainability. Got to do a little tour at a Chamorro village that was run by a lot of the indigenous folks from Guam. And they held up a coconut and they called coconuts travelers, okay? Because a coconut doesn't sprout unless it's still, okay? So a coconut that's being moved around and there's water or there's fluid inside the coconut. We all love coconut. Well, most of us, I hope, love coconut milk. I love it. I like anything coconut. But as long as that's moving around, the coconut won't sprout. And because of that, they called the coconuts travelers because the coconuts were able to colonize over millions of years all those islands throughout the Pacific as coconuts fell in the water and traveled around, and things hitched a ride on coconuts. Most organic things, right, most plants aren't able to do that. They're going to decompose long before they travel across the 620,000 square miles of the Pacific Ocean, right? So plastics are now providing a way for lots of coastal organisms that require some kind of substrate, a place to hang out, right? Coastal organisms are used to having some kind of structure, right? Because they're on the coast. Open ocean, like I said, right? If you're plankton, there's nowhere to hide. If you've got plastic in the water, you've got somewhere to hide, you've got some structure. And so one of the hypotheses in this is that the transport of plastics with all of their associated biota, all their associated living things are going to be a way to really move things around the globe that wasn't present before. And so the implication
Is that good or bad?
at this point, it's just a hand we're dealt, right?
We've seen the dangers of non-native species.
Yep.
Is there
That's what I'm talking
Yep.
about.
consequences to that?
There's probably going to be tremendous consequences. Yep.
We'll probably see new organisms.
So by the same token, lots of fish are now using these garbage bags. There's tons and tons of fish out there using garbage. Mahi is a fish that loves structure in the ocean, right? So in fact, poachers, put out big logs and things like that with GPS tracking. They'll throw it out, they'll go home, and they'll come back like a week later, and that's where they fish for lots of open ocean fish, because open ocean fish are looking for things like that.
That means we're getting plastics in our bodies. We're eating those.
Yeah, Mike Roman always does this trick where he holds up a credit card and says he's going to eat it, because the hypothesis is that we ingest about a credit card's worth about 4 grams of plastic a week, which is about how much a credit card is.
Oh, we.
So it's about 4 grams a week. Yeah. Now, to be clear, there's another study recently that came out. Well, there was a presentation at a recent conference that a friend of mine told me about, that they did some studies. So a lot of that plastic is coming from the processing. Okay, so if you think about it, if you go out and catch a rockfish, right? Rockfish season's coming up. They're right now up in the rivers, and they're spawning, and they're going to head out. Rockfish season's coming. You catch that. What kind of line are you using? Monofilament,
Yeah.
right? That's plastic. You're going to bring it onto your fiberglass boat. You're going to throw it in your plastic cooler. You're going to take it home. You're going to fillet it with a knife that probably has a plastic handle on a plastic cutting board. You're going to put it in a piece of Tupperware, right? So that fish has a lot of opportunities outside of living its life in the ocean to get plastic, right? There's a lot of potential for.
Yeah, but it's eating it when it's it's.
Yeah, but you're shoving it in a plastic bag and you're cutting it on a plastic cutting board. So, it's all of those things. It's all of those things, right? So,
So we're all,
Hmm?
X% of our body will be plastic.
well, yeah, I really hate to end on a bad note like this, but it is time for me to wrap things up. And again, I apologize I brought too many slides. I hope that this was really helpful. So, I'll just leave with these thoughts about plastic, that these are just my thoughts. So, how we've got to think about removing plastic, but we also have to think about energy and environmental costs of doing that, right? As I said, there's whole communities that have sprung up in the Great Pacific Garbage Patch, and if we just go reinvent trawling and scrape that plastic out of the water, we're also bringing lots of organisms out of the water with us. We got to think about the unintended consequences. Not saying we don't do it. I'm just saying we got to, for once in our human history, start thinking about the implications of everything we do. I think the easiest way to deal with the plastics is really to think about the source. Think about what we use for single-use plastics. Think about alternatives and think about recyclability. There's some new technologies coming out for recycling that I've read about, where you don't have to separate all the types of plastics. You can combine different types of plastics. They're actually engineering different microbes that are actually able to break down the plastics to their component parts. Plastics are polymers, meaning that they're multiple molecules all attached together, and if you can break them down to their original parts, you can reassemble them into new polymers. So it's a much more efficient recycling process. So there's a lot of people working on this. There's a lot of new technologies coming. Okay, the work that I've done has been funded by lots of different people and has been supported by lots of people. The thing I love about oceanography is that we've got a lot of really great people that work together on these problems. I can't do everything, right? I'm the plankton guy. I like to work with the physics guys, the geology guys, everybody else. So just noting that this work was supported by lots of people. And you can always reach out to me if you have other questions. And I hope that this was useful.